I'm just going to call it community continued.
Is it a continuation from the notes from yesterday?
Yeah.
Alright.
All right. So, number one, what I want to talk about are the cells and the molecules that are associated with the immune system. So number one, you read about it last night, this thing called an antibody. Okay? Antibodies are sometimes called immunoglobulins. Okay? And these are proteins. And the purpose of these proteins is to identify pathogens or antigens and either mark them so that they can be destroyed later by a phagocyte, or disable them, or at least alert cells that they shouldn't be allowing those things into their into the cell. Okay, so these are proteins that identify antigens and can mark them for destruction by phagocytes. Like I said, they do also do some other things. They can sometimes disable the functioning of those antigens on their own, and in other cases, they are just marking those antigens so that the cells know that they shouldn't be letting those antigens into the cell because they've already been marked by an antibody to tell them that it's a bad thing. Okay? So, the general structure of an antibody is something like this. Okay, they call this part at the bottom the heavy chain. And that's about all you need to know about that part of it. And then up at the top, you've got these things that are called antigen binding sites. Sometimes they're called antigen receptors. And the thing about an antigen binding site is every antibody is going to have a unique antigen binding site. So unique per antibody. Okay? So you can then expect that if there was an antigen that was like a blue circle here, that blue circle might bind to that antigen binding site, and it would be able to identify it, right? But if there was an antigen that was a red square, the red square would not bind to that antigen binding site, and it would need an antibody that had a red square antigen binding site. Make sense? All right. So let's talk about B cells. Cells produced in red bone marrow that contain surface antibodies. So here's a B cell. There's an antibody on its surface. Another antibody on its surface. Another one. Sure, that's enough. Okay, it got more than that, but that's enough for us to draw. Okay, so here's a B cell, and we're going to do 2 B cells. I'm going to cheat, but draw 2 B cells. And these B cells are going to be different from one another. And the way they're going to be different is that they are going to contain different antibodies. Okay, or different antigen receptor points or antigen binding sites. So this one's going to have blue ones. This one's going to have red ones. Obviously, they're not blue and red. I'm just trying to denote that they're different. Okay? So, let's say over here, this is, there's a muscle cell. Okay? From your body. And then the muscle cell on its surface has these red proteins. Okay, that means that B cell number 1 that contains the red antigen binding sites, if it were to come into contact with this muscle cell, it would identify the muscle cell as a pathogen. Is that good? No, that's bad. Okay, but this one over here that has the blue ones can bounce off of here, and blue does not interface with red, and therefore will not identify the muscle cell as a pathogen. That is good, right? So what happens here is something that is called self-selection. So self-selection destroys any B cell that identifies your cells or cellular products as antigens. Okay? So in other words, this guy right here is destroyed. This one over here is allowed to go and go to the spleen or wherever it's going to hang out in order to detect its pathogens, but this guy is killed off because he would have responded to your own cells as antigens, and that's bad news, right? So there are a couple different types of B cells, right? There are naive B cells. And naive B cells are the ones that they're the ones that you start out with as like an infant, and you have naive B cells for every single possible protein that's not present in your body, right? Because all the ones that were present in your body got self-selected and destroyed, right? And so there's not very many of each type, maybe like 4 or 5 of each type of these, and therefore it takes your body a little while for them to find the antigens. Yeah.
Can you explain why there's an X through the red?
Because this red B cell here would have responded to the muscle cell as an antigen, and you don't want your immune system to think your muscles are antigens.
Okay.
So it gets destroyed before it actually gets produced, or before it gets allowed to leave the bone marrow, and therefore only this one that didn't do that didn't respond to your own cells, was allowed to leave the bone marrow.
Okay, so the B cell gets destroyed?
Yeah, the whole B cell gets destroyed. Yep. All right. So you got these naive B cells. Like I said, very few of them. Their whole job is to identify antigens. Okay. When they do identify an antigen, they become active. Okay, and when they become active and become plasma cells, they can divide into 2 different types. Okay, a type called an effector B cell. And the effector B cell's job is to produce antibodies that are the same as the antibodies on its surface. Okay? And there's also memory B cells. Okay? And memory B cells are going to be able to detect future infections. Okay, so we said that the first time you get an infection, it takes your naive B cells a long time, a relatively long time to identify that infection, and so it takes you a while to mount an immune response, and therefore you get sick. You have some symptoms for a little while. Okay, but once you've been infected by that particular pathogen, your body creates thousands of these memory B cells, and the memory B cells are then there to make sure that that same infection is not able to really cause any symptoms for you again. Okay. It seems then that it would be better for us to just have lots more naive B cells. It seems like, you know, if the memory B cells can stop us from getting symptoms at all, why don't we just have like 5,000 of each of the naive B cells instead of having like 4 or 5 of them? Yeah. Okay, that's one reason. Wouldn't be able to produce as many red blood cells. What else? Yeah.
There's so many different antibodies and antigens that exist, so we need like an insane amount.
You'd need an insane amount. There wouldn't be room for them in your body. Your body, there'd be more B cells in your body than there would be any other cells, by far, right? And so that wouldn't work out. You wouldn't be able to do that. You'd be all B cells, right? And so what we have instead are these memory B cells. And the memory B cells are going to be a very specific subset of those B cells that are for your specific environment. So whatever you constantly get exposed to, the pathogens that you get exposed to on a daily basis, are the subset of memory B cells that you have. You still have these naive B cells in case you get some random disease from somewhere, but for the most part, you're exposed to a constant subset of viruses and colds on a daily basis, right? So the question then becomes about that is the subset the same everywhere? Like if I go to Africa, no, it's a totally different subset for them, right? They're still humans over there, but they're exposed to a totally different subset of viruses and antigens, and so I'm probably going to get sick if I go there. If you've ever heard that you shouldn't drink the water in Mexico, right? Anybody ever vacation to Mexico and they say like only drink bottled water, right? Okay, well why? Well, it's because you get horrible, terrible diarrhea if you drink the water. Why do they even have running water then? What?
It's fine for them.
They can drink the water. It's not a problem for them because they have memory B cells for all the nasty stuff that's in the water. And so even if they're drinking these antigens and constantly exposed to these antigens, they're used to it. They just have to get horrible, terrible diarrhea a couple of times, and then they have all of the B cells that they need in order to prevent that in the future, right? This is how this works. Does this make sense? Okay, so let's talk about T cells. T cells are cells produced in red bone marrow that are going to mature in the thymus. And there's going to be 2 different types of them. The first one is called a helper T cell. And helper T cell has a signaling protein or a receptor protein that allows it to interface with MHC2. Okay, so a cell that contains a receptor protein for MHC2 on B cells and phagocytes. So these B cells and phagocytes, those are the 2 cells in your body that are going to be looking for antigens and actively destroying antigens. Okay? And so the helper T cell's job is to make sure that the things that are being destroyed are in fact antigens. Okay? So these confirm antigens as pathogens. They're making sure that there's no rogue phagocyte or rogue B cell out there that's attacking your own body. Okay? And if they do, they're going to release these things called cytokines. To allow B cell division. Okay? There's another type of T cell called the cytotoxic T cell. And the cytotoxic T cell is going to, instead of reading MHC2, it's going to have a receptor protein for MHC1. Protein for MHC1. Okay? MHC1 is on all cells, all nucleated cells. Okay? And the job of a cytotoxic T cell is to confirm that no cells are making non-self proteins. Okay? So it identifies cells producing non-self proteins and destroys them. If it is able to destroy a cell, it's going to differentiate into memory T cells. And those memory T cells are going to detect future infections. And effector T cells that deal with the current infection. Okay? So, using these cells then, we say that these cells are what are involved in something that's called active immunity. Basically, active immunity is just the normal immune response. Normal, humoral, and cell-mediated immune response. We say that there's 3 types of cells that are involved. What's up?
Just like what's the order for like cytotoxic T cell? Like how does it get to memory T cells and effector T cells?
If it's able to destroy, so anytime it destroys a cell that's creating a non-self protein, it creates a specific effector T cell that looks then specifically for that non-self protein, right? And also memory cells at that time. It's both of these things.
B cell.
What's that?
It's the same thing with like the B cell that like.
Yep, exact same thing. Yep, yep. Except for it doesn't need a helper T cell to come confirm it. It just does it. Okay. Yeah.
Helper T cells help B cells.
Helper T cells help B cells. Yep. All right. So the cells that are involved in active immunity are going to be B cells, T cells, and also the phagocytes. And we're not going to take much time. Basically, this here is active immunity. This is all active immunity here. And then also this last part of this is active immunity, right? So those are all parts of active immunity. Okay, then you got passive immunity. Passive immunity is a temporary immunity caused by antibody presence or antibodies. The most common type of this is something that's called natural passive immunity. Natural passive immunity, the most common example of this is breast milk. Okay? So, breast milk contains the antibodies that are currently being produced by the mother. So whatever current antigens the mother is being exposed to, she is producing antibodies for those antigens, and then those antibodies get passed on to the infant via the breast milk. Okay? And this makes perfect evolutionary sense because theoretically, since the mother is caring for the infant, anytime the infant is going to be exposed to a pathogen or an antigen, the mother is also going to be exposed to that same pathogen or antigen because the infant shouldn't be anywhere where the mother is not. Right? Does that make sense? So, why though? Why is this good? Well, essentially what happens here in passive immunity is that the baby's immune system is all being handled by the mother, right? The mother is passing on these antibodies rather, and the antibodies are sort of solving any problems that the baby might have, solving any infections that that baby might have, okay? So we can say that this is antibodies are passed from mother to infant to prevent infections as long as the infant breastfeeds. Okay. So the key to realize here, though, is that these antibodies are being transferred directly via the mother's milk. But the baby itself has no way to make any of those antibodies. Because remember, the thing that produces antibodies are the B cells, right? And so the baby's not producing antibodies via B cells. It's not making effector B cells. It just has the antibodies because the mother is passing those antibodies along, okay? So if you've ever heard the phrase, give a man a fish and he'll eat for a day, teach a man a fish and he'll eat for a lifetime, you know that thing? Okay. So essentially what the mother's doing there is giving the baby a fish, right? Because the baby's got a lot of stuff to worry about at this point. The baby's like learning how to use its arms and legs and figuring out where its face is and like trying to figure out what all these noises and lights are and all the sensory inputs. It's got a lot of stuff to worry about. So it shouldn't have to worry about getting sick at the very beginning of its life. In fact, it's doing a lot of development that could be negatively affected by getting sick. Okay? So the mother then provides temporary immunity while that development is going on, but once that infant stops nursing, the infant is going to get sick because it's no longer being protected by all the mother's antibodies. Right? And it has to get sick. That's part of life. You have to get sick at some point so that you can produce all those memory B cells to allow you to not get sick in the future. Does that make sense? But it's essentially just giving this infant a small pass and saying, okay, you got other stuff to deal with right now, like learning to use your arms and legs, so let's let you not get sick at the beginning, and then later on when you're, a little older, you can handle being sick. You have a question, Amanda?
How are people born without immune systems?
What you mean?
Like there are those kids who have to like stay in a home all day.
There's bone marrow disorders. We'll talk about. Give me 2 seconds. We'll talk about those in just a second. Those are generally caused by autoimmune disorders that destroy the immune system. So it's not like they have no immune system. It's that their immune systems are faulty. Okay. All right. There's also something called artificial passive immunity. And artificial passive immunity is like medicine. Okay? And people who are immunocompromised are often given injections of antibodies in order to help them fight off specific infections. Immunocompromised. That means their immune system's not working as well as it should. Compromised. Can be given injections of antibodies to help fight specific infections. We'll come back to that in a minute to talk about something pretty cool. All right, but first, before we do that, let's talk about problems. Problems with the immune system, problems with immunity. First off, we'll start with the most common problem. What's up?
So why are the antibodies not rejected when they're injected?
Antibodies are sort of like universally accepted. In fact, basically all mammals have like identical antibodies to the point where like they're interchangeable. You can take the antibodies from one organism and put it into another, and like they'll do the same have the same function. They're just proteins.
Okay, just
Yeah.
proteins.
Yeah. Okay, so problems with the immune system. The most common problem, allergies. Okay, so allergies are a specific type of immunoglobulin, which is IgEs. Right, so they're caused by the production of IgEs in response to a false pathogen.
Mr. Tate, are we talking about like seasonal allergies or like?
All allergies, every type of allergy is caused by the same thing, and that is there's a B cell somewhere in your history that decided that some pollen or cat dander or food decided that was a bad thing, went ahead and divided into memory B cells, and then every time you get exposed to it, those memory B cells start producing these immunoglobulin E's. Immunoglobulin E's then bind to mast cells in your body, and the mast cells then produce histamine. Okay? So mast cells have IgE receptors, and produce histamine in response. So when you have allergies then, you would take an antihistamine like Benadryl or Claritin or one of those things, and that will prevent your mast cells from secreting too much histamine, and then will stop your symptoms. But you still have those B cells that are going through and creating those IgEs in the first place. Okay? This is really common, extremely common. Almost everybody has allergies to something, right? But less common are these things that are similar called autoimmune disorders. Okay, and autoimmune disorders refer to when your immune system decides that some cell in your body or some cellular product that you produce is an antigen. Immune system identifies a cell or cellular product as an antigen. So we generally divide these into three categories. one called inflammatory disorders. Basically what happens in an inflammatory disorder is you start out with non-specific inflammatory response, like, you know, something triggers a very small inflammatory response, and then your body freaks out about it and then creates an even larger inflammatory response, and it just kind of snowballs out of control. Okay? 2 examples of this are rheumatoid arthritis and another one that's much more common than arthritis are canker sores. So if you've ever had a canker sore, those things in your mouth, they're caused by an autoimmune disorder, a very mild autoimmune disorder where your body starts an inflammatory response and then sees that inflammatory response as an infection of itself and then triggers a even more massive inflammatory response that leads to you having a sore, right? These can be further divided into saying that you've got things like systematic disorders. Okay? A systematic disorder is something that is going to affect the entire body. Okay, so cells in all organs and organ systems and parts of your body get affected by these. A good example of this is lupus. Lupus is an autoimmune disorder where immune system starts attacking basically all of your organs, including, you know, your skin and your liver and your pancreas and basically, like I said, all of your organs are affected by lupus, right? And then you have targeted ones. Targeted disorders are only going to affect one specific part of your body. Only affect one cell or cell type. Okay? So, some examples of this are like type 1 diabetes. Type 1 diabetes is caused by your immune system attacking specifically the cells in your pancreas that produce insulin. And so those cells are seen as infected cells, as bad cells, and so they get destroyed by cytotoxic T cells. And as a result of that, your body can't produce insulin then because you don't have the cells that produce insulin. And so we tend to call type 1 diabetes insulin-dependent diabetes, or people who have to get insulin injections because their body cannot produce enough on its own, right? And then there's another one that's much less common, but I want to talk about it because it's very interesting, called acute aplastic anemia. Okay, and acute aplastic anemia is an autoimmune disorder that your B cells and T cells, or it's actually B cells or T cells, start attacking your bone marrow. Okay, so your bone marrow starts getting destroyed by your own immune system, and that's bad because don't forget that your bone marrow produces red blood cells, white blood cells, and platelets. So essentially, all of your blood contents are made by your bone marrow. Okay, so this starts going down and down and down. You've got less blood cells in your blood, and you start getting tired, you start getting sick. Lots of very bad things will happen. If you have acute aplastic anemia and it goes untreated, you will die, right? So the interesting thing, though, is how they treat it. And they use this treatment in a lot of autoimmune disorders. I just happen to know about this one because my dad had acute aplastic anemia, and so I did a lot of research on it. But it's very interesting and very cool. So the way that they treat it is, since your B cells and your T cells have essentially gone evil and they're starting to kill you, right? They take your blood out and they spin it down so that it's just your B cells and T cells left, okay? They get a bunch of them, enough that you have the ability to inject them into a horse, right? And they give a horse an injection of your B cells and T cells, okay? So essentially what you've done is you have infected a horse with your own B cells and T cells. And so the horse's immune system says, hey, we have an infection. We got to deal with this infection. And the horse's B cells then start producing antibodies that kill off your B cells and T cells, right? So then they take the blood from the horse and they spin it down so that now you just have a bunch of horse antibodies. And don't forget that horse's antibodies will now kill your B cells and T cells. And they give you, artificial passive immunity to your own B cells and T cells for a short period of time. Right? They start injecting you with what they call this serum, and they do it over the course of a couple of weeks, and they kill off every B cell and T cell in your body because, like I said, they've gone evil and they're trying to kill you, right? So you essentially go back to a state where you have no B cells and T cells, very similar to what an infant would have right after birth, okay? So then your bone marrow can start to regenerate, and your bone marrow starts to produce a whole bunch more of these naive B cells now, right? And the naive B cells will go in and do exactly what they do in an infant, which is, identify pathogens, albeit slowly, and then turn into memory B cells. So what you essentially have done then is you have hit the reset button on your entire immune system, and every memory B cell that you created throughout your entire life is destroyed. Every memory T cell that you created throughout your entire life is destroyed. It resets things like allergies. It resets any autoimmune disorder that you've had, but it also resets every memory immune cell that you've ever had, meaning you need to get sick again with every single pathogen that you've ever been exposed to your entire life, right? But you don't die, so that's a plus. What's up?
How do you acquire that?
Random chance is the best that they have come up with, right? So could be caused by some viral infection that causes, like let's say a viral particle gets into your a bone marrow cell, and then your body identifies that bone marrow cell as being infected, but then sort of incorrectly assumes that all your bone marrow cells are infected at that point. Random chance. Yeah.
If you have like a really severe allergy to like something really common, like that was really severe and could kill you, would it be beneficial to do this?
They certainly could do this. It would probably not be the best plan. There are much better plans for sort of reducing your response to allergies. One of them is by taking that allergen and constantly exposing you to low levels of it to the point where your body essentially gets tired of dealing with that specific immunoglobulin and just stops responding to it anymore. Like the mast cells are like, oh god, it's that immunoglobulin again. I'm not even going to release histamine this time, right? Because you're constantly being exposed to it. Yeah.
Is there any way for like a targeted disorder to be genetic? Because I know like a mom and both of her kids that have diabetes.
Yep. Absolutely. It's definitely got a genetic component, and arthritis has a genetic component as well. Rheumatoid arthritis does. There's a good chance that almost all these things have at least some genetic components because small changes in MHC1, which is the histocompatibility complex that every single cell has, can change the way that your T cells will respond to it. Yeah.